Good afternoon, everyone. Today, I would like to talk about the 2006 Nobel Prize in Physics. I chose this topic because it helps us understand how the universe began and how we can study its earliest state. The prize was awarded for the discovery of the blackbody form and anisotropy of the cosmic microwave background radiation. At first, this may sound very complex, but the idea is quite fascinating. Scientists believe that the universe began with the Big Bang. However, we cannot directly observe that moment. So how can we learn about the early universe? The answer lies in the cosmic microwave background radiation. This radiation is a faint signal that fills the entire universe. It is considered the afterglow of the Big Bang. Although it is very weak, it carries important information. Scientists discovered that this radiation is almost uniform, but not perfectly. There are tiny differences in temperature across space. These small variations are extremely important. They show how matter was distributed in the early universe. Over time, these small differences grew into stars, galaxies, and large cosmic structures. This discovery was very important. It provided strong evidence for the Big Bang theory. It also allowed scientists to study the origin of the universe with high precision. I find this discovery very fascinating. It shows that even very small signals can contain information about something enormous. It also teaches us that the past leaves traces that we can still observe today. In the future, this research may help us understand the origin and evolution of the universe even more deeply. Even though this topic may seem abstract, it gives us an important message. The universe itself is a record of its own history. Thank you.
Good afternoon, everyone. Today, I would like to talk about the 2006 Nobel Prize in Chemistry. I chose this topic because it explains how genetic information is copied and used in living organisms. The prize was awarded for studies on the molecular basis of eukaryotic transcription. In simple terms, this research explains how information stored in DNA is transferred into RNA. DNA contains the instructions for life. However, DNA itself cannot directly perform most functions in the cell. Instead, its information must first be copied into RNA. This process is called transcription. During transcription, a molecular machine called RNA polymerase reads the DNA sequence. It then creates a corresponding RNA strand based on that information. This RNA can then be used to produce proteins, which carry out most functions in the body. In other words, transcription is the first step in turning genetic information into action. This discovery was very important because it revealed how this process works at the molecular level. Scientists were able to understand the structure and function of RNA polymerase in detail. This helped explain how cells control which genes are turned on or off. I find this discovery very fascinating. It shows that life depends on precise information transfer. It also reminds us that even tiny molecular processes can control complex systems like the human body. In the future, this knowledge may help us develop new medical treatments. For example, it may allow us to control gene expression or treat diseases at the genetic level. Even though this topic may seem technical, it gives us an important message. Life is built on the accurate copying and use of information. Thank you.
Good afternoon, everyone. Today, I would like to talk about the 2006 Nobel Prize in Physiology or Medicine. I chose this topic because it explains how cells can control gene activity with remarkable precision. The prize was awarded for the discovery of RNA interference, or gene silencing by double-stranded RNA. At first, this may sound very technical, but the basic idea is quite clear. In living cells, genetic information is copied from DNA into RNA. However, not every RNA molecule should remain active all the time. Cells need a way to stop certain genetic messages when necessary. This is where RNA interference becomes important. Scientists discovered that double-stranded RNA can trigger a natural process that silences specific genes. When double-stranded RNA enters a cell, it is cut into small pieces. These small RNA molecules then guide the cell to matching RNA messages. As a result, the target RNA is destroyed or blocked before it can be used. In other words, the cell can shut down a specific genetic instruction. This discovery was extremely important. It revealed a powerful mechanism of gene regulation that had not been fully understood before. It also showed that double-stranded RNA is not just a simple molecule, but a signal that can control gene activity. This finding opened a new field of research. Scientists began to use RNA interference as a tool to study the function of genes. By silencing one gene at a time, they could observe what changed in the organism. This made it much easier to investigate how genes work. I find this discovery very fascinating. It shows that life depends not only on producing information, but also on controlling and reducing it when necessary. It also teaches us that very small molecules can have powerful effects inside living cells. In the future, this knowledge may help us develop more precise treatments for genetic diseases, cancer, and viral infections. Even though this topic may seem advanced, it gives us an important message. Life remains healthy not only by expressing genes, but also by silencing them at the right time. Thank you.